Hey everyone, welcome back. This is the second part of this bioenergetics lecture. So one thing to note is when it comes to the bioenergetics in our body and what's really going to give us the energy that we need to do things is going to be through adenosine triphosphate or ATP. And so this is a really important thing and you should really get familiar with because you hear ATP a lot because it's really the only usable source of energy in the body. And so its chemical structure, you can look at that if you're interested, but what happens is that our body is going to take the fuel that we utilize, so glucose, fats, things like that, and take a chemical called ADP, so adenosine diphosphate, and it's going to add an inorganic phosphate to that. So it's going to take an inorganic phosphate and ADP. It's going to combine those to make ATP. And so that's the process of using fuel to make ATP. And then what's going to happen is that ATP is going to go to where the body needs energy, and through something called ATPase, that ATP is going to be broken down into ADP and inorganic phosphate, and it's going to release energy so that things can happen. So think muscle contraction. You're using a bunch of ATP, breaking that apart into ADP, and that's going to allow you to produce movement. And so this is a really important concept to kind of go ahead and think about as we continue to move on through this conversation. And so here's that structure where you can kind of see, here's your 3 phosphates. So we're basically cutting off this last one and there's a release of energy. And so we're just adding it back and forth to take that energy, create it, send it somewhere so that it can lose that phosphate, become ADP, and that energy is released for use. And so you can kind of see here this conceptual of making that ATP and then taking that ATP over to use for another process, and realize the wheels get smaller because what happens is throughout we lose potential energy through heat. Heat dissipates and we lose that amount of energy that it can produce every step of the way, but it still allows us to do what we need to do. And so with that, we talk about these different systems. And so the formation of ATP comes through either phosphocreatine breakdown, which we'll talk about, glycolysis, or taking glucose or glycogen into ATP, and then oxidative formation of ATP, which uses things like fats, more so the byproducts from glycolysis, things like that. And so we break them down into 2 different pathways called anaerobic and aerobic pathways. Anaerobic pathways do not involve oxygen. They don't need oxygen at all to create ATP. And so this is your PC phosphocreatine system and glycolysis. And then we have our aerobic pathways, which requires O2. So the whole reason we breathe, to take in O2 so that we can use that to help make ATP. And this is our oxidative phosphorylation system that is considered the aerobic pathway. And again, that's oxidative phosphorylation. So with anaerobic ATP production, we have our ATP-PC system, so ATP phosphocreatine system. This is our immediate source of ATP. It is the quickest source that we have to take ADP and inorganic phosphate and turn it into ATP, and that is through a chemical called phosphocreatine. What happens with creatine kinase is it takes off a phosphate, adds it to the ATP, and then you get creatine and ATP. And most people are familiar with creatine because it's a supplement you can take, and it may supposedly increase performance, and we'll talk about that some this semester. But what happens is this is a very quick process. It makes a lot of ATP very quickly, but it also runs out very quickly. You can only hold so much creatine in your body, and once that phosphocreatine is turned into creatine, we've made that ATP. When you cool down after you stop exercise, this process will reverse. It'll take some energy and it'll reverse back into PC and ADP, which is how we build that system back up, but it does take time. And so this system's really quick, but as anything that's really quick and really strong that produces a lot of something in a short amount of time, gets tired very quickly. So in about 10 to 30 seconds, you've completely used all of that stores that you had of phosphocreatine. And so we need another way to make ATP. And so we have our second anaerobic system called glycosis. And what this does is it takes 2 glucose and converts them into 2 pyruvic acids or 2 lactic acids. And so those are the byproducts. We take glucose, turn them into pyruvic acid or lactic acid, and we get energy from this. But glycosis has two different phases. It has an energy investment phase where it's going to take 2 ATP, take 2 molecules of energy to start this process going. And then it has an energy generation phase, which is going to produce 4 ATP. So we put 2 ATP in with glucose and glycolysis, and we're going to produce 4 ATP. So we get a net of 2 positive ATP. Plus, we're going to produce 2 NADHs, which we talked about a little bit before, which are going to become important later on in the aerobic processes, and either 2 pyruvate or 2 lactic acids. It's pyruvate and lactic acid can go back and forth on what they are, and we'll talk about what that process looks like. And so here's lactic acid and what it looks like, and lactate. So if you hear lactate versus lactic acid, it's really just we've lost this hydrogen right here has come off. So the acid is because of the hydrogen here. When you lose that, it becomes lactate. Very quickly, it goes back and forth. And so here's another look at those energy investment versus energy generation phases, just kind of breaking it down so you can see that there's 2 there. And then the interaction between glucose and glycogen as fuel. They're both going to enter, and they're both going to do the same thing. They just enter at a little bit different step, and glycogen doesn't take quite as much energy to produce ATP as glucose does. So it enters in at one step further down the system. So here are all the steps involved in glycolysis, and I would say you need to learn this. It takes a little bit of time to learn all of these steps, but it is something good to know where the ATP comes in, where NADs are created, and where ATPs are created. But generally what happens is that glucose, you invest in ATP initially with, and what you do is you create glucose 6-phosphate. And then another enzyme occurs and you get fructose 6-phosphate. And then you have to invest another ATP right here to make fructose 1,6-bisphosphate. And so you can see it takes 2 ATP in this top half. And then what happens, you have an enzyme called atolase. And what that does is it actually splits this glucose molecule that's 6 carbons big into 2 3-carbon molecules that are exactly the same. And they both run through this same process. And what happens is you make, basically you lose a hydrogen in this first step, and you take NAD and create NADH, so you reduce NAD, and that's going to go and be utilized for energy later. And then what you're going to do is in the next step, you're going to take ADP and create ATP, so you're going to lose a phosphate group. And that's going to happen for each molecule, so you get +2 there. It goes and gets transformed a little bit. We lose some water off of it, so you can see how it gets smaller and smaller because it keeps losing pieces of itself. It's breaking it down. And then at the end, what happens is we will take and create another ATP with both pieces, and our end product is always pyruvate. So it always is pyruvate that we're going to make at the very end, but depending on what happens after this, it may become lactate or it may be used for more energy. And so you can see we've created 4 ATP, we've created 2 NADs, and our net gain is 2 because we remember it cost us 2 ATPs when we first started. So after that, what's going to happen is we have those ATPs, they're going to go to where they're needed and be utilized. But you may be saying, well, we've talked about NADs and you said they're going to create energy. Well, what happens to those? Well, they're going to get carried over to the mitochondria, the powerhouse of the cell. And this is where a majority of ATP generation comes from. And this is where our aerobic system happens. And so what will happen is we'll have things that will carry these NADHs over to the mitochondria, and pyruvic acid will more than likely get converted into lactic acid, especially in an environment where there's not a lot of oxygen. If there is a lot of oxygen, it'll get moved into the mitochondria as well and be utilized for energy. And same thing, if we create FADs through other processes, those would get moved over. And kind of goes through the process, letting you see chemically, structure-wise, where that goes. LDH stands for lactate dehydrogenase, how we convert pyruvate to lactate. And so eventually what's going to happen is we're going to move into that aerobic ATP production. So if you're doing something for a longer period of time, those glycolysis and ATP PC systems can't keep up. they get tired, they run out of products, and so we have to kind of move over at about the 2-minute mark of constant exercise. We start relying more on this aerobic energy system, and it doesn't produce as much ATP as quickly. So it's a slower system, but it goes for a lot longer. And so it's really broken up into two kind of parts with the aerobic system. And the first step is called the citric acid cycle, or Krebs cycle. You'll see it as Krebs, Krebs. And then the second part is the electron transport chain. And so the Krebs cycle completes the oxidation of fuels from fats, and our pyruvate will go into this proteins if we need them to. And then the electron transport chain will take those NADs that we create, we'll create some in the Krebs cycle as well, and it will transform those into ATP as well at the very end. So you can look at Hans Krebs and his discovery of the citric acid cycle. I think it's great to kind of just get some background on that. I'm trying to see if this is still talking about him or if we're moving kind of into it. So what happens is, we'll pick up here. So steps leading to oxidative phosphorylation. So glycosis takes a 6-carbon sugar molecule, gets 2 net ATP out of it, breaks it down into 3-carbon. And then what happens is that 3-carbon pyruvate from glycolysis goes to the mitochondria, and it's going to lose another carbon, creating more CO2 that we're going to breathe out. So we're starting to see where that CO2 production comes from. And it's going to create acetyl coenzyme A, which is going to allow this citric acid cycle to start making more ATP and NADHs. And so this talks about that process a little bit more. You can feel free to pause and kind of read through that. Hopefully you're reading in your book as well. That basically we keep making something to then kind of grind it down, tear it apart so that we can create ATPs and NADHs and FADHs. And then at the end of the citric acid cycle, that just continues to be circular. It just keeps going and going as long as the products are there for it to keep moving. Literally circles around on itself. What will happen is those NADHs will go to an electron transport chain. And those NADHs will move across a membrane layer. And in doing so, they basically store up energy on one side of this membrane. And then when they release it, it causes the creation of ATP. And that ATP, we have some byproducts from that creation. We have some hydrogens left over, and we want to get rid of those because those aren't great for our body. And so it'll take a piece of oxygen that we've breathed in and create a water molecule. So the whole reason we're breathing is to get to the mitochondria, get that oxygen in there. And at the end of this electron transport chain, when we've made ATP, we're going to attach some hydrogens to oxygen and make water so that we can then continue to not lower the pH of our cell. And so, again, it keeps talking about this, and it's really good to kind of focus on this. I don't need to kind of walk through and read every line. You will see something about GTP being created. Realize that GTP will eventually transform over to ATP. So it's a different protein structure. So it's guanosine instead of adenosine. And so it's the same thing, and it will eventually convert over to ATP. So if you see that there's a creation of 1 GTP, that's the same as saying we created 1 ATP. So here's the actual Krebs or citric acid cycle. So pyruvate starts the process. It loses a carbon and becomes acetyl-CoA. And then it starts moving through this, and each one of these are citrate is the molecule, and then actinase is the enzyme that is going to change citrate into isocitrate. And then isocitrate dehydrogenase changes it to alpha ketoglutarate, and you can see in that process we created some CO2, which we're going to breathe out, and we're going to take an NAD and create an NADH. And so this process continues on. We create 2 NADHs over here as it moves through. We create a GTP, which will be converted into ATP. Over here on succinate to fumarate, we create an FAD to an FADH. And again, we go fumarase to maltate, which creates, and then maltate to oxaloacetate, which creates an NADH. And then what happens is oxaloacetate will, with citrate synthase, combine with acetyl-CoA that we created from pyruvate and start the cycle back over. So for every turn of this kind of cycle, what you produce is 3 NADHs, 1 FADH, and 1 ATP. This is the aerobic system, and it does take glucose utilization. It does take the glycolysis. Byproducts have to be present, pyruvate, to make acetyl-CoA to keep this system going. If any one of these systems isn't there, if we are lacking in any part of these enzymes or substrates like citrate, what's going to happen is the cycle will stop. And so that's really important to kind of note. And so with each pyruvate we put in, we talked about the products we get out. So if we take one glucose molecule, run it through glycolysis, we get 2 pyruvates, and so that means we would get 2 turns of this. And so we can start figuring out how much energy we can create from one molecule of glucose, which is really nice because we can start seeing how, well, if glucose is able to go through the aerobic system, we get another ATP out of it. We get some more NADHs, and we'll talk about how many of those, how that creates energy and how many per. So realize when we talk about fats, they can come in at any point in this system. It can add some more maltate. It could add more isocitrate depending on how they're broken up. Typically, the way it's done, though, is fats will be broken up into acetyl-CoAs. So you have through beta oxidation. The glycerol is an important muscle fuel during exercise, so that can be a big thing. But acetyl-CoA is the main thing. So we'll take our fats and break them up into two carbon molecules of acetyl-CoA and kind of keep this system going too. So it doesn't have to start with glucose, but it can, and it likes to be mixed in with that as well. Protein breakdown can come kind of anywhere in those intermediate processes, not the biggest thing. And so you can see the relationship between metabolisms and fats. They're all kind of interconnected a little bit from where we're getting these from our triglycerides, our stored fat, to our glycogen from glucose and proteins, and what part of this process they're going in on. There are preferred spots, but it really just depends on what is needed and what's going on inside the body. So electron transport chain is still aerobic, and it is part of the system. It's kind of thought as the last kind of area that it goes, but it's going on at the same time. Realize all of these processes are occurring all the time. Oxidative phosphorylation occurs in the mitochondria as well. And what happens is it takes NADHs and FADHs and it produces ATP. And what happens is for every 1 NADH that's produced, you're going to produce 2.5 ATP. You can't really produce half an ATP, but if we put 2 of these together, you would get a total of 5. So through their products. FADHs, you get 1.5. And so what you can see is you can start adding up through one glucose molecule how many total ATP it is. And if we look at the fact we created, I want to say 5 NADHs and 1 FADH, plus we created 5 total ATPs, we start making a lot of energy molecules for one piece of glucose. It did cost us a couple at the beginning, but we can do that math. And here's that statement again of using O2 to make some water to form that ATP with those. Let's see. So what happens is basically the hydrogen from NADH and FADH, this H at the end, the hydrogen is moved across the membrane and it creates an electrical gradient through this pumping action. And then when they move back through to the other side, that electrical energy is turned to combine the ADP and the inorganic phosphate into ATP, which is how that ATP is created. And so you can kind of see how it all kind of starts flowing together, and you're getting this ATP production. Realize a fat molecule, we've kind of talked about glucose, but a fat molecule on this, a fat molecule can be many, many carbons long and can be cut up into hundreds of 2-carbon acetyl-CoAs, and for each one of those, it's going to run the citric acid cycle one time. And for each of that, you're going to get 3 NADHs, an FAD, and an ATP. And so you can see that there's a lot of energy that can be stored in fat, depending on how many times it's going to go round and round. Here's a theoretical example of ATP, the electron transport chain. There's 4 pumps, and each one is moving hydrogens over through basically some activation, and then at the end those hydrogens come back down and produce ATP and some water. Your book should do a good job of walking you through this process to get a basic understanding, and I think we'll talk a little bit more about it this semester as well. So all of these electrons being stored up here, sometimes they do leak out. They get out in other places besides through the pump, and so this can create something called a free radical. Free radicals can damage cells, and so this is kind of where this is thought to happen. So when you exercise, these pumps are utilized. They become a little bit more efficient. Kind of everything flows a little bit better. So we get a reduction in these free radicals when these systems are used. So people that don't exercise tend to have more free radicals because there's a buildup of these electrons because you're not needing that energy production, and they tend to slip out and make them that cause damage. So here's kind of that grand total from one molecule of glucose, how it runs, and through each of these processes, what is created, and at the end, what is the total ATP that's added up to. And so you can kind of see that grand total from one glucose molecule. We can make 32 ATP, which is really great, and then it only cost us 2 ATP to start that process off. And so it gives you some more numbers that you can kind of work with and show you how moles and the chemistry portion of this, and realize that a lot of our energy efficiency is pretty low, and that a lot of the energy we make is lost to heat. There are rate limiting enzymes to these steps, so they can only process so quickly as it is. The more of that rate limiting enzyme we have, the quicker it can go. But that kind of sets the pace. If that is at a low level, then the amount of ATP you can create will not be as high as it could be if you had higher levels of it, because it's the slowest process. Basically everything, think about a stoplight, right? If the stoplight is red, no one can go through. When the stoplight's green, people go through. And then after the stoplight, maybe traffic speeds up even more. So it's kind of like that slows you down kind of thing in that production area. Each system has its own rate limiting enzyme. These are really good to know. And so knowing that creatine kinase is the rate limiting enzyme for ATP PC system, glycosis, it's something called phosphofructokinase. So knowing kind of where that is in that system, but each one has its own rate limiting enzyme. And so if these get too low, it's going to affect the end products, the ATP production. Hydraulic model, so just kind of look at that a little bit, not too terribly important for what we're talking about. But at the end of the day, the big thing to kind of remember as you're talking about all this is all these systems are going on at one time, but the one that's contributing the most ATP at the time is going to be based on kind of the duration and intensity of exercise. And so this is saying long-term low intensity is going to be a majority through aerobic. Short-term high intensity is mainly anaerobic. And then things in between are going to be a mixture of the two. And so it's really good to keep that in mind as you're thinking about these. It's not one or the other. It's all of them at the same time, and which one is the brightest light bulb in the room, kind of like they're on dimmers. This is a good chart, kind of shows you activities based, like take a look at this, the activities and which one percentage is more aerobic or anaerobic, and the durations that those may take, because this kind of would make a difference in how you were interacting with someone on what they were doing or what their goals are, what you may need to focus on as far as energy systems go. So that finishes our lecture. If you have any questions, let me know. I hope you guys are doing well with the information so far, and we'll talk to you later.